Okay, thank you everyone to be here. It was sticking here during this break. As we said, my name is Filippo Utro. I'm a senior researcher in IBM Research in New York, and I can see from the title of this presentation, Transform Healthcare and Life Science with Biomedical Foundation Model and Quantum Computing, we have quite an ambitious goal. And you can imagine a lot of different background and science has to be brought together to achieve this goal. And we know that we cannot do this alone. So for this reason, during the year, we built several partnerships, several collaborations with different institutions all across the world, academia, other industry, and so on, to build a meaningful scientific impact. And you can see there some of the recent journal where we publish some of our papers. What's of these many collaborations I want to mention? One is in particular that we have recently started with Cleveland Clinic. And the aim of this collaboration is to accelerate discovery in drug in different diseases to try to understand better the disease, find biomarker, find new possible target, new molecules that could help the patient, the clinician there. And we try to achieve that with two different main pillars. One is through AI. The other one is through quantum computing. What you see in that picture over there is an actual quantum computer, which is sitting inside Cleveland Clinic. And you can see from the picture, is what you will think is in a very safe environment from or outside from anyone else, is actually being installed in the cafeteria. So you can see there people having their coffee while quantum computing is running some of their experiments. So the first one of the two pillars that I want to mention that we are working on is through AI. And you can see that AI clearly evolved over time. We started from the machine learning, which was a concept that, if you think about, was born around 1960, but only later in the late 90s, early 2000, we actually found application to it in life science. More recently, that notion of machine learning, we moved to deep learning, and today we have foundation model. You can see there how changed drastically the different background in science, different applications from both life science and healthcare, as well as NLP and other contexts, which we use almost every day, sometimes without even knowing. So the first one, what I want to say, is actually this change of paradigm, where before in machine learning, we were thinking to select a specific data set, we curate the data set, we try to train, work with the specific data set, create a model, validate it, and then we finally release it. With the foundation model, instead, we start with a large amount of data from different sources. We then try to build a model and then fine-tune it with the specific use case. So what we are trying to do now in IBM is actually creating a biomedical foundation model with that integrates different modalities from small molecule, omics data, we're coming from genomics, transcriptomics, and so on, to actually build three different novel models to identify and discover targets, small molecule, to better understanding some of the biologies behind that. We're planning to validate those models through both in silico and in vitro through our collaborators. So here, I'm not going to go into the details of each of the model. I'm just going to give you a generic idea of the model which is behind, and as well as some of the results we have with that, some of the publication. The first one I want to mention is this one, where we show how AI can be used to, this was done in collaboration with Dr. Fei-Xiong Chang from the Cleveland Clinic, where we show how AI can be used to discover a possible drug for Alzheimer's disease. So the first model which I want to talk about is the small molecule, which can be, which has very specific goal, as you can imagine. It can be used to do different downstream applications, such as generation. So you can generate new possible molecule with a specific profile. The other one is to prediction. So given a possible molecule, what will be its phenotype? And clearly, you can think about, you can use that one back to another. You generate something, and then you try to figure out how it works out. And the model takes an input to possible different data sources as well, and a different way to include the molecules, for instance, such as 3D structures, mile sequencing, possible imaging, and so on. Here we show a result on application of our model. for instance, in active site sequencing representation. And you can see the details of the 2 publications related to it there, if you are in case you are interested in that. The other model which I want to show is the one we call biologics. The aim of this model is to create a component for biological therapy discovery with the goal of leveraging large-scale representation of protein sequencing structure and dynamics and for diverse downstream task association for multiple biological modalities. This model can serve as the basis for different downstream tasks, such as in therapeutic design. You can think for generation and assessment across antibody, vaccine, and so on. Here you can see one of the recent results using this model, where we were applying an HLA-A2 peptide immunogenicity. Finally, we have the last of our model, which is the BMFM target. We actually have 2 different models. One is the genome model, which uses SNPs and GWAS data for disease risk assessment, estimation, and gene disease association. The other one instead using single-cell RNA-seq for cell type annotation and gene perturbation prediction. In summary, we have this set of biomedical foundation model, which we're trying to also combine also as well together to accelerate the discovery using the new AI technique. And as was mentioned before, we know we cannot do this alone. So for this reason, recently IBM has launched an AI alliance, which incorporates together with Meta, over 50 institutions across the world. And you can see some of them listed here. Now, I want to switch gear and move about paraphrasing a bit the Bob McDonald bestseller around about what is the future, although the future in some sense for quantum computing as well is now. So you can see that IBM has a long history in quantum technology. And clearly, as you have seen the keynote from Dr. Warren at the beginning, where she was showed the small computer that she was using at the beginning of her research without even an hard drive or anything, and how you can think how that technology span across over time, how long it took to generate the computer that we have today, what the machine we have today. If you think instead the quantum computing, a few years ago, we were working on quantum computing that had only an handful set of qubits. Now we have quantum computing which over 100 qubits. So they do not follow the law more, the development of quantum computing, and that speed is way much faster with happened to classical computing. So now I want to show some of the applications that we are using quantum computing in life science. So we decide to, first of all, of course, you don't need quantum computing for every single problem, right? There has to be a problem where classically you have a sort of bottleneck in terms of accuracy or in terms of performance, which you want to then use quantum computing for. So we decide to take a cell-centric therapeutic view, and we consider 4 different work streams. The first one was in CAR T-cell optimization or micromodel tumor environment, perturbation of single-cell level, and as well as the 4th one, which actually can be used as a basis of the other work stream, which actually to try to capture high-order interaction between the interaction between the data that we have. So, of course, there is many models for a technique for quantum computing. They do a very specific task. So I'm just, for the reminder of this talk, I'm going to focus only on the specific task that we have been using in our work. So the first one is about the genetic engineering for CAR T cell. So my colleague tomorrow is going to give a deep dive talk. So just to give you an idea of what's going on in this work stream and just spend a few words, the CAR T cell T therapy, it's a very interesting problem where you have a library of motifs. So you can think as an alphabet, let's say of size 14, and you need to place in certain position this motif to create a sequence. And those sequences can go usually between 4 and 5 position, and you can already think about if you have 14 possible motifs in 4 positions, you have a combinatorial space of around 2,400 possible combinations, which clearly you cannot experimentally analyze all of them. Only a small portion can be used. Let's say 250 in our experiment, in our work, we've been using around 250 data points. And classically, it's been shown that the best model starting from this load regime data to predict all the remaining space, possible combination, it's around 70% of accuracy. And this most likely due because the low quantity of data. And this low data regime plus the bottleneck in accuracy of the classroom is where maybe quantum can go and help and provide some real benefit out of it. The second one is the microtumor environment. So you can think as well as quantum can be used to enhance embedding space. You can see that very nice in that cartoon where you have your data in a classical computer. The embedding is not optimal because it's not separating the blue from the red. But when you go in a quantum computer, the embedding can be maybe separate the data very well. And this can be very effective when we talk about, in the Hilbert space, when we talk about cell and tissue representation. So the quantum can then be used to capture hidden relationship and correlation spatial in time in the data. And then for this reason, could actually be used for for single cell spatial transcriptomic, where classically you can map this problem to a graph. So each cell will represent your nodes. Interaction, you can put a cell next together, and then you can annotate the node with information such as possible mutation, expression, and so on, characteristic of the cell. The first classical model to solve this problem is the one of using graphical neural networks, so a GNN. But the GNN is very computational demanding, and in particular also suffers some very limitation when you want to query your data or analyze the data directly in the embedding space. So what instead can be used, you can combine and create an hybrid approach using a quantum machine learning together with the GNN. So in this figure, I'm showing, for instance, how the GNN can then be used and fed a quantum machine learning approach, which can use the embedding space to then improve the performance of the GNN or found some of the optimal parameter for it, and go back to feed again the GNN to output the solution. And this loop can be reiterated many, many times until we found the solution, the desired solution. Next, I'm going to show you another application, which we have in two. The first one is about single cell perturbation model. Recently, it's been shown optimal transport. They're very good in performance this task, but they still have some many limitations of how that can be used and what are the performance, the preservation of the model itself. So we think that quantum optimal transport can then be used to improve the performance over the classical one. Finally, I want to move on to our next task, which is about capturing complex interaction in the treatment of disease. So this actually can be seen both at single level to the population level. As you know, the disease sometimes is often the results of multiple factors going together. You may have multiple endophenotype that then lead toward the phenomenon of the disease itself. So, and because the disease do not work alone, right? Not single cell, not environment works alone. So I found and captured those relationships is quite very hard. So topological data analysis, and in particular cumulant, could be used to capture this. But when you talk about 2, 3, 4, 5 order relationships, so consider 5 elements together, you can already imagine now the combinatorial space is exploding, right? It's also very difficult from computation and memory-wise to analyzing those. And this can be clearly transmitted at the single cell level. You can think of you have a single cell, and then maybe consider to apply a drug, or using, for instance, in cell line, the impact of a drug and measuring the effect, which, for instance, you can go and take it from that map. And you want to see how, going back, what's the relationship, what's the biological relationship between the targeted versus all the other one, and maybe find possible dual relationship or synergy between 2 different genes that impact the cell line, right? So, or multiple one again. So again, finding those interactions can be very complex and high demanding. That's why we think that quantum computing can go and help solving this problem. So again, before in many of these our tasks and what I just mentioned, it was then also put on our prospective paper in collaboration with many institutions and many co-authors all around the world. You can see them, list them there. And you can also see some of our recent work in to cover many of these aspects there. Right, so And as I mentioned again, as it takes a village, we're also building a community around quantum computing for healthcare life sciences. So if you're interested, please let me know. We are having a series of working group around different aspects from clinical trial, possible biomarker, QML application for omics, and so on, that we want to engage people, experts in the community, both life science, quantum, computer science, and so on, maybe to discover and see how quantum can be used and be helpful for those applications. So finally, I just want to highlight some of the talk of work been having here in ISMB. Some of them are being already passed, unfortunately. So we're before this talk, but as mentioned, we have some extra, like tomorrow morning, about our application of QML for CAR T-cell engineering. And I will be happy now to take any question if you have any.
Thanks, Filippo. Any questions from the room? No? Any online? Oh, that was AstraZeneca. So I guess I have a general question.
Mm-hmm.
So what do you see happening in the next 10 years in quantum, just with your background?
I see happening a lot. I see happening in different levels. The first one will be, for instance, on hardware, where hopefully we're going to get larger machine with better noise, so quality of the qubit and so on. So that will allow us to directly use larger data as well as better model for it. That's one. The other one, I'm assuming they also will give a series of applications in different contexts, not just life science, but as well economics and so on, which they are a very top topic right now.